examples of blood-based biomarker investigations in migraine. Starting with my disclosures briefly, and then moving on to my first slide. So there are a number of different key properties that we're interested in the context of a blood-based biomarker, bearing in mind that although the title of the presentation is blood-based, this can relate to biological fluids such as saliva as well. A good biomarker will help us with diagnosis, will correlate with disease severity, and it might be able to predict or track treatment response. A blood biomarker must differentiate between healthy and disease and between diseases. And in order to achieve any of these things, the biomarker must give us accurate data. That's absolutely crucial. So the overall goal of today's presentation is to provide an overview of some key information needed to interpret and develop studies involving blood biomarkers. And I'm going to give a number of examples. These are not exhaustive by any means. I don't have time to go into everything. And this isn't necessarily anything about recommendations of a particular approach, but they're things that are really important to bear in mind in the context of biomarker studies. This provides a summary of factors to consider that I will go over, which start with study design and biological considerations within the patient, then the collection of the sample and handling and its processing, which are pre-analytical considerations. And in addition to that, you have analysis and interpretation considerations that I won't have time to go into today. But the point from this slide is that these factors all come together to make it very challenging to identify robust biomarkers. And if we think about CGRP as the example that I will use throughout this presentation, bearing all of this in mind helps us understand why CGRP currently isn't a robust biomarker for migraine, even though we think perhaps it might be. So study design and biological considerations then. So the first thing to think about is where is our substance of interest, our analyte, and I've got some definitions in these orange boxes throughout the presentation. Where is our analyte being produced, and how does its site of production and its concentration at its site of production relate to the site at which you ultimately take the sample from? An endocrine hormone, for example, is produced in a gland and is directly released into the blood. So this will, in principle, have a reasonably high concentration in the blood at which you are sampling. Whereas a neuropeptide, for example, like CGRP or PACAP, being produced in a neuron to act locally, the amount that you see in blood is more likely to be a spillover from the neural site. So at the neural site, its concentration might be high, but by the time you're taking a sample from the blood, it's effectively diluted. It also becomes diluted because as soon as the substance has been produced, there are biological mechanisms to clear it from the system in order to maintain sensitivity to that substance. If we look here at this example, where we have an infusion of CGRP into human subjects to reach steady state, as soon as that infusion is stopped, you see a rapid clearance of that CGRP from the blood. And this is caused by a combination of mechanisms, which are hepatic or renal clearance, accounting for up to 50% of the loss of CGRP from the blood, receptor-mediated clearance, and also proteases. So effectively, you have further dilution of your sample as soon as it's been produced. And so what we're looking at here is that our sample taken is only a snapshot of this continuum from production to clearance. Receptor-mediated clearance, we don't know exactly how much of this is at play in the case of CGRP, but we do know that this does occur in that CGRP binds to its receptor, and once that receptor has been activated, the receptor is endocytosed or internalized. The CGRP that is bound to the receptor then becomes degraded by endosomal protease-mediated degradation. So the receptor is returned to the cell surface without the CGRP. So you have a means by which CGRP can be cleared via the receptor. And this can occur for other receptors as well. You also then have protease or peptidase-mediated degradation. So many peptides, like CGRP, often have enzymatic cleavage sites. These red arrows indicate sites of enzyme recognition within the human alpha CGRP sequence. And we know that incubation of the synthetic peptide with different enzymes can produce degradation and formation of fragments of the parent peptide. And those fragments are then not expected to be active, so another means of controlling the biology. But what we aren't sure about is how many of these actually occur in vivo, but it's something to bear in mind. We then have the design of the clinical study itself in the context of the points that I've made so far, which is which site at which do you take your sample from? Are you better off taking your sample from a site that is closer to the expected site of release than a more distant site? So, for example, is jugular venous blood in the context of migraine or saliva potentially more informative than cubital vein blood, where you might have greater dilution of that sample and therefore more difficulty measuring it? You also have to bear in mind your sample timing relative to your hypothesis, given those clearance mechanisms I described. If you hypothesize that your substance, your analyte, is released during an attack, do you need to measure that sample at the peak of the attack? But practically speaking, that may not always be possible, so you have to weigh these things up in terms of your particular study. There's blinding to bear in mind always, and then the sample size relative to the expected variation. How high is intra-patient variability versus inter-patient variability? And there are studies that suggest that within a patient, the variability is not necessarily that high, but between patients it is. So might you be better to consider repeated measure sampling and appropriate statistical analysis, where you measure individual patients between or within attacks to improve the quality of your data. Once you've designed your study and you've factored in these points about clearance, there are these pre-analytical considerations to take into account when actually taking your sample and then processing your sample in order to get good data. Starting with which blood collection tube or which collection tube do you use in the first place? Now, as you are well aware, there are many different types of potential blood collection tube for a number of different biological matrices that can be produced from that. And just to define what I mean by biological matrix, this is, of course, the discrete material of biological origin that can be sampled and processed in a reproducible manner. So we take blood, and from that, the matrix is either the blood, plasma, serum, or it could be saliva, for example. And each of these different biological matrices has different properties, and each collection tube has different additives. And each of those additives can potentially create interference in the assay that you wish to perform. And so these are important factors right at the beginning when you initially take your sample. Two very common blood-borne biological matrices are, of course, plasma and serum, but it's really important to bear in mind that these are not interchangeable. And the suitability of each analyte within each matrix must be rigorously tested. So just as a reminder, plasma is, of course, derived from blood that's been centrifuged after collection, where you have anticoagulants in your tube. So you take the plasma fraction from your centrifuge tube, whereas serum is processed differently, where there are no anticoagulants because serum is derived from after clot formation. And this, in practical terms, results in a delay between cold collection and cold storage. Your plasma will be taken and centrifuged rapidly, and then you can take your plasma fraction and store it, whereas serum needs to sit on the bench for approximately 30 minutes for clot formation. If you have proteases that are active in your sample, busily breaking down your peptides such as CGRP, you could imagine that serum may not be appropriate for certain molecules. And in fact, the protein complement that is in each of these sample types is different. And so you need to think carefully around the suitability of that matrix for your particular analyte in question. This is also important where the matrix itself can have an influence on your assay. This is an example of this, where we're looking at the measurement of CGRP in an enzyme immunoassay, and we're just looking at standard curves. In the enzyme immunoassay buffer, you see a nice close to linear standard curve here. But if you were to prepare the same standard curve in 100% human plasma, you see quite a starkly different profile, where the measured concentration is substantially lower in the pure plasma. And this is because you have a matrix effect where there is apparent interference of the plasma on the measurement of the substance. This also results in a hook effect or prozone, where increasing analyte concentrations result in no change or decreased signals when compared to the preceding concentration. If you dilute that plasma, you return to almost 100% recovery. And this is by no way a recommendation of anything in particular that you might do, but it's something to bear in mind, that you should always be considering how you prepare your samples in terms of how you do your assay and what influence the matrix itself might have on the outcome of the experiment. In addition to your blood collection tube, you also have other factors to bear in mind in terms of sample processing, what other additives you might add, and the exact type of tube in which you perform your experiment. So protease inhibitors are worth a mention. As I indicated, peptides like CGRP can be broken down by different proteases, and many people therefore automatically choose to add cocktails of protease inhibitors to their samples. However, we have to bear in mind that protease inhibitors can interfere with assays. And in the results that I showed you previously with the CGRP standard curves, if you add certain protease inhibitor cocktails to those samples, you completely lose your measured CGRP counterintuitively, because the protease is in fact interfering with the assay. So addition will not guarantee better results, and in fact they could be worse. And one of the issues is it's hard to know exactly which proteases are most active in vivo. So this needs rigorous testing for each analyte in each assay. Depending on your analyte, you also have to factor in whether it might adsorb to the plastic that you're using in your tubes. So many peptides like CGRP have a net charge, which causes them to adsorb to surfaces. And that's discussed in this paper here. So you also just need to bear in mind whether you're going to lose your sample onto the plastic throughout the course of processing your sample. Then how do you store your sample? How stable it is, and how do you handle it? So stability and benchtop stability refer to the intactness of the analyte under the long-term storage conditions, say in the freezer, or as you're handling the sample on the bench. If we look at some example results here for CGRP, we're looking at a spiking 500 picograms per mil CGRP into 25% human plasma in enzyme immunobuffer. If you look at the fresh sample that's been immediately spiked, you see almost 100% recovery. And interestingly, multiple freeze thaws of that sample don't appear to affect the recovery. In contrast, if the sample is stored at different temperatures, this has a substantial impact on recovery. The fresh sample, again, is giving close to 100% recovery, but 4 degrees storage for 24 hours results in almost 100% loss, and room temperature for 6 hours results in approximately 90% loss. So this recovery, which relates to the extraction efficiency of the analytical process and is reported as a percentage of the known amount of analyte carried through the sample extraction and processing steps of the method, is important. So you do need to measure explicitly the recovery of your sample through spiked sample analysis whilst validating your assay. And this particular study from the Messlinger group has done a number of these things recently, and this study is certainly worth looking at for some of their recommendations with regard to the particular assay that they were using. And that's an important point. Each assay will have specific different requirements because they are all different. So that does bring me now to assay type. Do you have a radioimmunoassay? Do you have an ELISA? Or are you doing mass spectrometry? All of them have different properties and different considerations in regard to the points I've made so far. They will have different upper or lower quantities that can be accurately determined with acceptable precision and accuracy. They will have different sensitivities, they will have different selectivities and different specificities. So selectivity and specificity are particularly important when you're considering that many assays rely on antibody-based detection of a particular analyte. Because any antibody-based assay is critically dependent on what the antibody detects. So many different assay antibodies are often raised against or detect a part of the full sequence. So if we take CGRP, for example, this is a 37 amino acid peptide. Many suppliers will produce antibodies that are directed against only a portion of the sequence, say the C terminus. So if your assay detects something in your sample, it's detecting this portion against which the antibody was raised, not necessarily the full-length sequence. And so what you don't know is whether you're detecting only a fragment that contains that sequence or the full-length peptide. And that's where some assays which involve two antibodies, a sandwich ELISA, where there is an antibody directed against both the C terminus and the N terminus, can be more accurate if you wish to measure the full-length peptide, because when you see a signal, it can only be because you have both being detected. Another factor to consider is whether you have cross-reactivity with your assay antibodies. And this is particularly acute when we consider CGRP versus Amylin, because they are close to 50% identical in amino acid sequence. So that means if an antibody is raised against the C terminus here, there's a high chance that you could see cross-reactivity because you have a lot of similarity between those sequences. And this does in fact occur. Amylin antibodies can detect CGRP. So this is a dot plot looking at the ability of a particular amylin antibody to detect amylin, and you see positive detection. But also note that you see detection of CGRP. And this can also occur the other way around, where CGRP antibodies can also detect amylin. If you look at these numbers, you might go, oh, well, 10 micrograms, that's a lot higher than the amount that it's detecting amylin. And that's true. Its relative ability to detect amylin is much greater than CGRP. But you have to then bear that in mind in the context of biology. If we consider a neuron, it has very high amounts of CGRP. So even if you have just a small amount of cross-reactivity of an anti-amylin antibody with CGRP, there is still a high chance that it will detect CGRP, because whether staining is present is a balance between the quantity of the antigen and the level of cross-reactivity of the antibody. So in summary, the quantity of a biomarker or analyte measured in a biological sample is a snapshot of the continuum of its levels that will vary according to the relationship of the sample to the site of release, the interplay between receptor and organ-mediated clearance and protease-dependent degradation, and methodological factors including how the study is designed and how samples are processed. So overall, I would say for things like CGRP or say PACAP, there are currently more pitfalls than pearls. However, if we bear in mind these points about study design and biological considerations, our pre-analytical considerations, and then our analysis interpretation, if we bear all of these points in mind to design and validate and report and interpret our biomarker studies appropriately, I think this will help us move forward in the field. Just to close, I've provided some further reading and guidance documents. In particular, this top source here in orange is the source of most of the definitions that I've given to you. And if you are conducting any biomarker-based studies, if you're developing any methods or even buying an assay from a supplier, you should bear these points in mind. So that's the end of my presentation today. Thank you for listening.